Hi everyone, I'm Rizal Karim, a PhD candidate in the University of South Florida. I hope you guys are all enjoying a sunny summer as we always do in South Florida. Today I'll tell you a short story on epigenetics and how I'm trying to discover new drug molecules targeting this epigenetics. You have all heard about genetics and genes, which we inherit from our parents and cannot be changed during our lifetime except getting some diseases. Epigenetics is similar to genetics in a sense that we also inherit this from our parents, but unlike genetic information, which are stored as DNA sequences, epigenetic information are stored as a slight modification of histones and its surrounding proteins. Thus, this epigenetics add an additional layer on our genes. Interestingly, research in epigenetics show that functions of a gene can be changed not only by some diseases, but also by our actions such as exercise, our food choice, medicines, and many more. This additional gene control mechanism of epigenetics makes it much more interesting. So, how does this epigenetics work? In the epigenetic process, a first set of enzymes changes the chemical group that is attached to the DNA or its surrounding proteins called histones. These chemical groups are usually very small, such as hydroxyl, which is an oxygen and a hydrogen, or a methyl, which is a carbon and three hydrogens. After that, a second set of epigenetic reader protein recognizes these chemical modifications and control the functions of genes in our cell. Unfortunately, like any other process in our body, this epigenetic process could go wrong and can lead to a number of diseases. Now the question is, can we tackle those diseases using epigenetics? That is the question I'm investigating in my research. To understand the epigenetic mechanism in molecular details, I'm studying how a class of epigenetic reader protein called the bromodomain protein recognizes a specific chemical modification on histones. In my research, I have crystallized a number of bromodomain from the purified proteins and determined the 3D structures at a scale that is 1/10 of a nanometer. This method is called the protein crystallography. From the structures of these bromodomains, we see that their shape is very similar, yet individual bromodomains can recognize a specific modification on some certain histones. At the atomic level, this recognition works as if like a key can open a certain lock. Through this mechanism, bromodomain proteins control the function of genes, which could lead to certain diseases that we can definitely prevent by inhibiting the function of these bromodomains. Importantly, in several blood cancers and solid tumors such as pancreatic cancer, breast cancer, it has been found that these bromodomain proteins are overactivated, and more importantly, inactivation of this bromodomain protein has been shown to be beneficial so that it can stop the progression of cancer growth. Therefore, I'm discovering new small molecules or drug molecules that could interrupt the function of these bromodomains to control the disease progression. For this purpose, I have tested hundreds of chemical compounds against these bromodomains and have discovered two promising molecules that can bind to these bromodomains as we see in the 3D structures, and these chemical molecules works as a key for some specific two bromodomains. And most importantly, these chemical compounds can prevent the progression of cancers in some certain cancer cells. So I'm very excited about this discovery, and I hope that further work on these chemical compounds will give us some new drug candidates in the future. Thank you very much for your time, everybody.